The first descriptions of a plant cell appeared in the 17th century, after the invention and improvement of light microscopes. In the middle of the 19th century, German scientists Schleiden and Schwann formulated a theory about the cellular structure of all living organisms, which substantiates the main provisions. All living organisms are built from cells. There are unicellular and multicellular organisms. The development of any multicellular organism begins with the unicellular stage. Due to the improvement of microscopic technology, by the beginning of the 20th century, all cellular organelles were described. And since the middle of the 20th century, with the advent of electron microscopes and new technologies, it has become possible to study the cell in even more detail. The science that studies the cell is called cytology. In the late 19th and early 20th centuries, German scientists Fechsing, Reichner, and Haberland for the first time tried to cultivate an isolated cell in a test tube in support of the idea that a plant cell could give birth to a new organism. The study of tissues is the subject of the science of histology. The more tissues in the composition, the higher the organization of the organism. This is the basis for the idea of the evolutionary development of the plant kingdom as a whole from simple to more complex and larger organisms. Botanists have long known that the most important tissues of vascular plants are combined into three systems: dermal, vascular, and ground tissues. Ideas about the possibility of plant cell structure appeared in biology in the middle of the 19th century. American biologist Robbins and his colleague, the German histologist Kotte, discovered in 1922 that meristematic tissues of roots could be developed in a synthetic environment in the laboratory conditions, and in 1932 the American White and the Frenchman Cotre showed that the meristems of the root are able to grow indefinitely with the replacement of nutrients. By 1960, several methods for obtaining large masses of cell suspensions had been developed, based on the cultivation of a simple cell and a cell protoplasm, as well as clonal micropropagation. They are widely used in plant breeding. In recent years, cell and tissue research has received a new impetus, and the rapid progress of cell engineering technologies continues.